we're very near to dinner now, so forgive me if I skip some of the details of the gene changes, but I'll give you some idea of the work we've been doing recently, and it involves resistance training, strength training in rodent models. So I just want to acknowledge my co-authors. Take that. Thank you. Yes, so I'm at Liverpool John Moores University. Mark Viggers is now at the University of Florida. Chris Cardozzo, who Ashraf also works with, is in the Bronx, of the VA in the Bronx. And my colleagues in Vienna were very influential in making the implant that I shall talk about the use of. Doesn't this work? I don't know. Okay, so the context: a strong, healthy musculature is an important part of general health through the lifespan. This afternoon, we've had that in mind. Complex training programs are mainly the result of trial and error, to be honest. There is a huge experience in elite sport, where coaches think that they know the best way to train any sort of athlete, but mainly their experience, which is true and helpful, is mainly based on trial and error. And if you're dealing with a patient, or if you're dealing with an elderly person, or maybe a non-elite athlete like you or me, some of you might be elite athletes, I don't know, but then we need better evidence. We need evidence-based principles for guiding exercise. So the sorts of questions we try to answer is more always better. That's what you would learn from the internet. You need to exercise more and more and more. Well, that's not always true, especially for patients with muscular dystrophies, let's say. Okay. Do you always need to exercise to failure? You'll hear that. How can we avoid injury and drop out in youth coaching? I think that's a very important question. How do we stop damaging our young athletes? Why do some persons seem to respond so much better to a training program? How do we train without damage? How do we train in the elderly? How much exercise is just enough? Can we find circulating or salivary or urinary markers that will help us to know whether we have exercised effectively? Well, they're all questions that need mechanistic answers, and our rodent models are designed to try to make some progress there. So our main model is what I'll talk about today anyway, is resistance training. That means strength training once per day, or yes, in one of experiments, it's not once per day, but we use an implant. So this little implant can go inside the abdominal cavity of a rat or under the skin of a mouse, and the electrodes deliver activation to usually the common peroneal nerve and the tibial nerve of the lower hind limb. So our resistance training model is that in one limb, we only stimulate on one side, so the other side is a good internal control. Yep. We activate both the ankle plantar flexors and dorsiflexors together. So tibialis anterior and EDL that lift the foot are activated fully, maximally, and then we activate the plantar flexors to provide a resistance to the dorsiflexors. So this is loaded contraction. We analyze the tibialis anterior muscle in all of this, the work that I'll talk about now. So I had a reminder from Ugo this morning that that idea of co-contraction or agonistic contraction across the ankle in our case is very much like was used in the RISE project, where we had big electrodes on the quadriceps, which also activated some of the hamstrings so that there was co-contraction across the knee in that case. And in some of the cross-sectional CT scans, you can see that both the hamstrings and the quadriceps in those denervated patients were increased in cross-sectional area. So it's a similar approach that we're using to that was used in the RISE project. Okay, we are interested in the training time course, not just a single time point, but a time course of training, because exercise, the effect of exercise takes some hours and days and weeks. So in this study, the studies I'll talk about now, we make the implant, we allow a period of recovery, then we are able to start the stimulation, the daily stimulation, and then we harvest groups of muscles at 2 days, 10 days, 20 days, and sometimes 30 days. So we're looking at a time course. There's no need for daily supervision of the training because the implant is fully automatic, autonomous. It delivers the stimulation as we require it. And the implant actually keeps a record of the stimulation. So we've already published that with this training once per day in rat, we get about 15% hypertrophy. So here is muscle mass rising after 10 days, certainly, then between 20 and 30 days, it reaches a new plateau state. So we get reliable hypertrophy with this co-contraction model. So it's 5 sets of 10 repetitions once per day. Okay, typical strength training. I'll just have a drink. So I'm trying to go fast. We sent samples from the trained muscles after 2, 10, 20, and 30 days. And this is for RNA sequencing. So this is principal components analysis of those transcriptional changes in the stimulated muscle. And you can see that the control muscles on the unstimulated side separate clearly from the 2 days, 10 days, 20 days, and 30-day clusters. So there's clearly a progressive transcriptional change. And I'll come back to this, so I won't go through in detail now, but you can see that, of course, when you do RNA sequencing, you're overwhelmed with data. But in the top 500 differentially expressed genes, we could see patterns of change over the time course. So these little graphs show 2, 10, 20, 30 days. I know you can't see that, but you can see some genes rose and then declined again over the course of training. Some of them rose and declined more slowly. Some of them rose a little slower and declined. Some of them rose and stayed pretty high. And there was a group that declined and then stayed low. So there's a big change in the muscle gene expression. That I should have emphasized that all of the muscles were harvested 1 hour after their last exercise session. So remember, it was training just once per day, but these changes in gene expression are 1 hour after the last exercise session. So the point is that there's a big change in gene expression, and it changes over the time course of the exercise. So the change in the acute gene expression, the exercise response, is different after 2 days to what it is after 10 or 20 or 30 days. The muscle adapts the muscle is adapting to the daily exercise. Okay, so that was published in the FASEB journal last year, and we made that point. The acute transcriptional response changed along with progressive adaptation of the muscle phenotype. And actually, there's just been a preprint available from the Hanshin lab in Basel, which makes the same point for endurance training in mouse. This is in mouse, actually. They say the training status determines the acute exercise response, very similar to what we said. So you can't think of the exercise response as a fixed response. It changes as the muscle adapts to training. All right, so that daily training in the rat caused hypertrophy, a 15% increase in the mass, but it also, as we found, caused changes typical of endurance training. So the NADH levels were up. The muscle actually was a little bit redder. Do you see this is the stimulated muscle sliced up like sushi, and it's redder than the unstimulated muscle. Okay, sorry for any vegetarians here. And one critical thing that we presented at last year's Padua Muscle Days was that we saw this substantial decline in fast myosins. Last year, I said it was 2B and 2A, which came down. Actually, it's only 2B. We made the mistake because the rat annotation of the genome is not as precise as the mouse. So we thought that 2A was coming down, and it wasn't. It was only 2B. Okay, and so this is the accurate data. Myosin type 1, the black lines are rat. 2A doesn't change very much. 2XD doesn't change very much, but the 2B does come down with the daily stimulation. So in a way, you would say that was a model of if you were seeking only hypertrophy, daily stimulation was too much, okay? Because we're seeing some slowing of the muscle. If you were a power lifter, that would matter, yeah? Because you don't want your muscles to slow down. Okay, so we tried exercise in another group of rats every 3 days. And you might know that that is the recommendation for human exercise. If you want to strengthen your muscles, you read the recommendations from the American College of Sports Medicine. They'll say exercise once every 3 days. And even people who go to the gym every day, they usually only exercise each muscle group, say their upper body, once every 3 days. Well, surprisingly, the rat fits the human recommendations very well. We actually had a sham group in this case, so the sham and the control were very, very, very close together. But then we had daily stimulation, which is these two here. It was only a small group, but then every 3 days also separated out. And here are the results for the hypertrophy. And you can see this is the everyday group, the open bars, and that's the 15% hypertrophy everyday stimulation. Every 3 days stimulation, if anything, caused slightly more hypertrophy. So here's a case where you can exercise only a third of the total input gives you the same hypertrophy. Okay, and just as an aside, that level is the amount of hypertrophy you get if you don't use the co-contraction technique. If you don't load the muscle, you only get a very little hypertrophy. So in our model, loading is important. Okay, so just I'll go very briefly over this. This is a comparison of control versus 30 days daily stimulation. There's the extreme downregulation of 2B myosin. And up here, you can see the top gene pathways are to do with oxidative phosphorylation, so endurance type changes. When we change to every 3 days, you see much less downregulation of the fastest mycins. There's myosin heavy chain 4. And the top gene pathways that are modified don't include oxidative phosphorylation. So every 3 days, this resistance training gives more of a pure strength training stimulus in this rat model. Okay, I know I need to speed up. So we don't need to say that. Okay, so that was all in rat. But most mechanistic work, as you've heard this afternoon, on muscle growth and atrophy is done in the mouse. So we miniaturized our stimulation system for reliable use in the mouse. So what happens in the mouse? If we use exactly the same stimulation, daily stimulation, what happens in the mouse? I could make a quiz, yeah, a poll, but I won't, I'll tell you. Is it the same or different? That's the question. No, it's not the same. Okay, there's no. Okay, the mouse is used for many studies in mechanisms of muscle adaptation. So there's a good model from the Yan lab where mice have to squat to get their food. There's a good model from the Hornberger lab where mice are trained to drag a heavy cart along a track. And in those cases, they both see some hypertrophy in the exercised muscles, but they need a long time and a lot of contractions. I think, here we are. So in the Yan lab, they saw about 9% increase in mass, but they reckon their mice were squatting at least 200 times a day. So really a lot of exercise. Every time the mouse fed, it was doing resistance exercise. In the Hornberger lab, that paper has 15 weeks of training, and they reckoned that it took one of their members of staff half of their time to train a group of animals. So it's a very intensive method. Okay, and the transcriptomics in those papers are endpoint studies, and we focus more on the time course. Well, here's the answer. With daily resistance training in the mouse, we saw no hypertrophy. So there's the rat increasing in mass. With just the same stimulation, we saw no hypertrophy, no increase in muscle mass in the mouse. So the big question is, does the mouse not make an exercise response to this daily exercise regime? Well, on the contrary. So let's look at the 10 differentially expressed genes that we know overlapped. Let me show you. In our FASEB journal paper, we made this analysis where we overlapped 5 sets, including ours, 4 other sets of transcriptomic data with resistance exercise. And these were the 10 genes that were upregulated or differentially expressed in all 5 sets. I won't go through those in detail. We can talk at dinner. But for those genes, let's look at the time courses for rat and mouse. And when I saw this data, I was amazed at how similar the pink and the black lines. So the pink is the mouse, the black is the rat. So for those 10 genes, 9 out of the 10, this one is the outlier. This one is the only one which is really different between the mouse and the rat. So for all of these other genes, you can see that the time courses are extraordinarily similar. It looks as if my postdoc made up the data. It almost looks too good to be true. I assure you he didn't. But so the exercise response in terms of the transcriptional response is incredibly similar, but there's no net hypertrophy. So the differences must be surely important. So we're going to be looking carefully at the differences between the expressed genes in the rat and the mouse, because they must be influential in the genuine hypertrophy that we see in the rat. Well, if you, yeah, I'll have to skip that because it will just take too long. But we in the rat data, as I showed you, we classified the changes in genes into 5 classes by self-organizing mapping. And so we've looked at the 5 different classes and the top 10 genes in each class. And you can just see that the mouse and the rat data is really very similar in all of those classes. I've just gone through and selected a few genes like this one here, where it's obvious that the mouse is responding differently than the rat. Or in this case, no, they're pretty much all the same there. Anyway, the ones that I have just picked out with a very crude analysis as being different between rat and mouse include KLF6, HABP4, myoglobin, AMD1, which is involved in polyamine biosynthesis, this translational initiation factor, EIF4E3, the 2B myosin changes in the rat, but it doesn't change in the mouse, PGM1. So this is some first clues as to where the differences lie. If we look on purpose at genes involved in remodeling, so here's FOXO1, myostatin, FBOXO32, and TRIM63. Well, FOXO looks different in the mouse, hardly changes. Myostatin, there's a rather different response. So yeah, maybe these are clues that this exercise stimulus in the mouse isn't driving remodeling to the same degree as it is in the rat. And this is actually the first sort of unbiased comparison. Here are unique regulated stronger responses in mouse, and they tend to be related to the proteasome. And in the ones that are upregulated more strongly in the rat tend to be related to the TCA cycle. So we're beginning to see a pattern. But most of the genes respond very similar. So here is a correlation between 1,217 common features, and you can see the fold change in the rat is really very similar to the fold change in the mouse. So we can have a near identical exercise response, but hypertrophy in one case and not in the other. Okay, so we present a time course of changes in the transcriptomic response to identical exercise over a period of adaptation. Only 20 minutes of day per day cause 20 minutes of stimulation per day causes a substantial hypertrophy in the rat and near complete suppression of the 2B myosin. Resistance exercise every 3 days gives just as much hypertrophy as exercise every day. So the rat is a good model to look at overtraining effects, in my opinion, and its periodicity, the fact that 3 days gives just as good hypertrophy as every day, suggests that the rat may be a good model to translate to human periodization of exercise. There aren't existing time courses from human data, or at least the ones that are there don't give this level of detail, but I think they soon will. I think people will soon be doing similar transcriptomics in human muscle, and I look forward to that. The transcriptomic response is remarkably similar in mouse for most genes, including adaptation, but without increase in muscle mass. In vivo models of exercise response are remarkably concordant with human, lots of overlapping genes, and give the opportunity to look at the adaptation of mature, innervated muscle. I want to emphasize that. I recognize that we must try to replace animal experimentation as much as we possibly can. But those of you who work with culture systems, I know we get great mechanistic information, but for now, I think we do need to carry on using animal models of mature innervated muscle. So, a couple of limitations, but thank you for your attention. Amber has a question.